Hi, and welcome to an overview of Chapter 3 from Bauman, Microbiology, Cell Structure and Function. We'll use these next couple of slides to review the typical parts of the prokaryotic and eukaryotic cell. At the end of covering Chapter 3, we should have good mastery of the following learning goals, which will become very important as we move on into some of the later parts of this course. The differences between different kinds of prokaryotic and eukaryotic cells really becomes essential for two things. Number one, understanding that cells are different enables us to more easily be able to identify an unknown microorganism. And second, the differences between a pathogen and human cells enables us to potentially target therapies that will attack and eliminate the pathogen before damaging the host. This is a principle that becomes very, very important as we go into understanding about chemotherapy, this idea of selective toxicity of chemical agents. Now, any discussion about cells and different kinds of cells needs to give an overview of some of the basic properties of cellular life. The original idea of the cell theory was developed by this guy, Robert Hooke, back in the 17th century. And he's generally acknowledged as being the originator of the cell theory of modern biological thought, the idea that all living cells need to arise from pre-existing cells. All cellular life has a number of characteristics in common, which you can see delineated here in these bullet points. First, all living cells will grow and take up nutrients and get typically larger in size. Now, this doesn't happen endlessly because all living systems, cellular systems, also typically will have the ability to reproduce where they copy their genomes, and one cell can form two identical daughter cells in the process. Cellular life also typically demonstrates the principle of responsiveness to things in the environment. A cue or trigger out in the environment, let's say maybe something like heat, can cause the cell to do something, perhaps move or change its properties. Cells are very good at being able to respond to things out there in an environment. And then finally, metabolism is a property that living systems also have. They will take up nutrients from the environment and use those nutrients in controlled chemical reactions inside the cell, and therefore maintain a constant internal environment, this principle of homeostasis. Cellular life is broadly broken up into two classes of cells. We'll talk about each of these individually and then spend a fair amount of time talking about the specific parts of each of these different classes of cells. The first type of cellular life we're going to talk about are prokaryotes. We can see a number of characteristics that all prokaryotic cells have in common. First off, all cellular life uses DNA as the basis of its genome. Prokaryotic cells do as well. However, when we look at this DNA genome that's found in every single prokaryotic cell, what we find is that DNA is contained within the cell, but not within a organized structure that contains a membrane. As we'll see when we talk about eukaryotic cells, that membrane-bound DNA structures are the primary characteristic of the eukaryotic cell. Prokaryotic cells also additionally lack all sorts of internal structures with membranes as well. They tend to be very, very simple within the cytoplasm of the cells, and as a result, typically prokaryotic cells are going to be rather small in comparison to other cells. We see that they're approximately 1 micrometer in diameter, though this may vary upwards to about 10 micrometers of diameter. As we'll see, we use this size characteristic as really one of the first things that we might bring to fold to be able to figure out whether or not a given cell is a prokaryote or a eukaryote. In terms of the types of organisms that make up prokaryotes, it's an extremely large domain of life. We find that there's two broad classes of organisms that have prokaryotic cells. These are the bacteria that we'll spend a lot of time talking about in this course, and the archaea, which superficially, under the microscope, very, very closely resemble bacterial cells. They very much resemble bacterial cells in their internal structures and their size and so on. But when we look at them from a genetic basis and a biochemical basis, we find that they're actually very, very distinct from bacterial cells. we're probably not going to spend very much time in this course actually talking about archaea. Bacteria are very significant human pathogens. Archaea are very, very important from the standpoint of the biosphere, but as far as contributors to human-based disease, they're not particularly significant. Eukaryotic cells are going to be very, very different from a prokaryotic cell. As we indicated in the previous slide, all cellular life shares this property of having a DNA genome, but the DNA genome that's found in a eukaryotic cell is enclosed with a membrane, and this membrane-bound structure containing DNA is known as a nucleus. Eukaryotic cells additionally have lots of other membrane-bound structures within them. These are referred to as organelles, and as we will see in the last part of chapter 3, these organelles carry out specific roles within the cell. And we'll talk about the individual roles that these things play. Eukaryotic cells are typically much larger than a prokaryotic cell, and that may be our first clue. If we see a very large cell under the microscope, odds are it's going to be a eukaryotic cell. Inside, they have a much more complex structure with many more parts serving these various roles. The types of organisms that have eukaryotic cells, we can see a listing of these classes of organisms. The algae, protozoa, are both primarily composed of single-celled organisms, although the algae do have many multicellular organisms as well. And the fungi, the animals, and the plants also composed of eukaryotic organisms, though they are going to be primarily multicellular. Now, as we saw when we talked about the different types of organisms that make up prokaryotes, bacteria and archaea superficially very closely resemble one another, and for an untrained microscopist, they may have a very difficult time being able to tell the difference between bacteria and archaea. Even a novice biologist will have a pretty easy time telling the difference under the microscope between these different classes of eukaryotic cells. The fascinating thing to me actually is that although these different classes of eukaryotic cells, the algae, the protozoa, fungi, and so on, look very different, from a genetic standpoint, they're actually remarkably similar to one another. And so it's very, very interesting and fascinating evolutionary biology description that comes out from understanding these. Here we see a couple of examples of different kinds of cells, and our first clue as to what each cell might be is actually given to us by the size of the organism. Look, for example, at these orange cells up here in the upper left. We can see the length of these cells is probably approximately 2 micrometers in diameter. That's a good indication that the organism up here are prokaryotic cells. Over here in B, we see actually 2 kinds of cells, these and this much larger one. We can see that the larger one in diameter is probably going to be somewhere on the order of around 15 to 20 micrometers in diameter. This is a neutrophil. It's a human white blood cell. This red blood cell is also a human cell, but you'll note that it doesn't have a nucleus, and in fact human red blood cells are the only cells in the human body that lack a nucleus. Over here in C, we see a very long cell. If we were to gauge its length, we'd probably guess that it was probably on the order of around 200 micrometers in diameter. This is almost certainly a eukaryotic cell. And then finally in D, a humongous organism. Looking at the diameter of this, we would guess that this cell right here is probably on the order of around 10 millimeters in diameter, which would certainly be easy enough to see by the naked eye. Now, as we stated, our first clue for deciding whether or not something's a prokaryote or a eukaryote is going to be its size. And eukaryotic cells are typically going to be significantly larger than a prokaryote. And it becomes an interesting question to ask, why might this be? Well, it turns out that eukaryotic cells can actually get quite huge. The largest eukaryotic cells are typically going to be oocytes or eggs, and this ostrich egg you can see is 13 centimeters in diameter. That's pretty big. You see some other cells over here, a protozoan that's about 14 micrometers in length. We saw some cells that were about that big in the previous slide. And your typical bacterium only about 1 micrometer in diameter. Well, it turns out that the reason that eukaryotic cells can get bigger is because they're able to get around one of the problems that physics imposes upon them. And that has to do with the ratio of the surface area of an object to its volume. Why is this important? Well, it turns out that as something gets bigger, the surface area surrounding it doesn't increase at the same rate that the volume does. And that ratio of surface area to volume does not increase in a linear manner as something gets bigger. When this becomes important is as something gets bigger, that larger object is going to be limited in its ability to take up nutrients that it needs in order to grow. And it's going to be limited in its ability to get rid of waste products that it generates as it grows. How do eukaryotic cells solve this problem? Well, it turns out that a eukaryotic cell actually has all these internal membranes and organelles that, in essence, effectively increase the surface area of that cell. We'll see how that works in this cartoon in this next picture. This table simply demonstrates that as your surface area begins to increase and your volume begins to increase because the size of the cell increases here from a radius of 1 to a radius of 10, the surface area to volume ratio does not increase at the same rate. We can sort of see this illustrated right here. If we imagine 2 perfectly round cells, all the nutrients that this cell has to take in order to get bigger have to come in through the surface, through the outer membrane of this cell. Because this cell, this larger cell, has a smaller surface area to volume ratio, it can't take up nutrients as quickly or as rapidly as the smaller cell can. So nutrients entering into a cell with no internal membranes, what that means is that the bigger cell down here is not going to be able to grow as quickly. It's going to grow much more slowly. Well, what happens if we have these internal membranes? We see that this larger cell here with these internal membranes that I've drawn in here, what this does is it effectively increases the surface area available to take up nutrients. What that means is that more nutrients can enter this cell than this one. And if more nutrients can enter the cell, it means that the cell is going to be able to grow at a more rapid rate. And the growth rate of that cell is going to be increased. All right, so that's a basic overview of the different kinds of prokaryotic cells. What we'll spend in the next part is talking about the different types of structures that we find in the prokaryotic cell, working our way from the outside of the cell to the inside of the cell. Some of these structures that we see up here are going to end up being optional, and some of these structures are going to end up being required. And we'll talk about each of these different structures in turn.